Hi, everyone. We're going to start with Chapter 7, where we now are transitioning into looking at carbohydrates and glycobiology, the biology of sugars. So we're finally transitioning out of proteins and into a new type of biomolecule, one of the other main biomolecule building units in most biology, particularly in mammals, which we focus on, but also in plants, of course, and that is the carbohydrate. So here we're going to introduce kind of the basic building block of the carbohydrate or sugar, structures and naming of those building blocks, just like what we did with amino acids for proteins. I also want you to work on learning naming and structure of the basic units of sugars. And then we're going to talk about some of the ways that we visualize and look at forms of sugars, their properties when they start to chain together, just like, again, with amino acids, chaining together to make proteins, our building blocks of sugars will chain together to make long polymers of sugars. And then we'll kind of look at some examples of those types of sugars that are polysaccharides and their other forms where sugars can bind to other biomolecules. So carbohydrates are named based on their chemical formula. Anything that has a chemical formula with C, so carbon to the N, that means some number of carbons, so an N number of carbons and an N number of H2O. So carbohydrates are all just made out of carbon, hydrogen, oxygen, and they, again, are going to be carbohydrates if they fit this formula. They are produced from CO2 and water in plants, right? Photosynthesis is one of the main known types of processes that make sugars, and that occurs in plants. And of course, we as consumers of those plants will take in sugar and then break it down. Sugars and carbohydrates range in their size. It's a huge range. Again, similar to proteins, you can have something that's a small single unit sugar. It's as small as the smallest one, glyceraldehyde, which only has a molecular weight of 90 grams per mole. Or you could have a huge polymeric sugar that's made up of a bunch of subunits in molecular weights that are greater than, for example, here, this would be 200 million grams per mole. And that's something like amylopectin. So again, large diversity here. Sugars also fulfill a variety of functions. So one of the main ones that you are probably familiar with is as a source of energy. So energy sources and storage of energy is a main use of carbohydrates, but they also are used for structures in our cells and in our organs. There are structural components to cell walls and also in exoskeletons, for example, in certain organisms. Lastly, and this is really interesting, they can also be used for information. So they can be used to send information or to give information in a signaling process. So this is when our two cells or a cell-to-cell interaction occurs. For example, you might turn on a process of metabolism, and sugars can sometimes be used as the key, essentially, to turn on or off processes in signaling. Carbohydrate nomenclature, the naming of carbohydrates, is generally very simple. You use the number of carbon atoms that exists in the carbohydrate and then add the ending "-ose". So for example, if you had a 3-carbon long carbohydrate, it would be triose. Something that is a 6-carbon carbohydrate would be a hexose. So we're using those same types of endings that you're familiar with, probably from organic chemistry when we were naming carbon chains, and you just add the ose ending. Now, carbohydrates can also be broken down into their type of functional group, their main functional group that they have, in particular a carbonyl functional group. Remember, a carbonyl is this type of group, a carbon double bonded to an oxygen, and there are a variety of types of carbonyls depending on what is attached to this carbon. So for example, if there are 2 more carbons on each side, this is called a ketone. And if there is a carbon and a hydrogen like this is an aldehyde. If you had, for example, instead a OH group here, this would be a carboxylic acid. So again, these are all types of carbonyl groups, right? The main point is that they have this carbon double bond oxygen, and there are a variety depending on what's bound to that. So again, review those functional groups, but in this case, for sugars, there are 2 types of main functional groups that sugars initially have, and that is either an aldehyde group or a ketone group. If it is a aldehyde type sugar, it is called an aldose. And if it is a ketone type sugar, it is called a ketose. So to name something all together, we can look at this example. This is the main carbonyl group on a sugar, and as you can see, it is an aldehyde. So we know that this is an aldose. We also know that there are 3 carbons in this sugar. So altogether, this is an aldo triose. The common name for this particular sugar is glyceraldehyde. Now, I am going to be going through a lot of common names, and I have given you a few charts, and I expect you to know the general idea for naming sugars, which is this. But there are a set of sugars that are very, very common, just like our set of amino acids that are utilized very heavily in biological organisms. And I expect you to memorize what they look like, their structure, and their name. Here's another example of a carbohydrate here on the right. You can see that the main carbonyl in this case is instead a ketone because it has a carbon on each side. So it is a ketose. And in this case, it also has 3 carbons. So this one is a ketotriose, and its common name is dihydroxyacetone. Glyceraldehyde and dihydroxyacetone, actually, if you get into metabolism and in Biochem 2, you'll see that they are a main component of the breakdown of other sugars in our body during glycolysis, which is one of our main pathways of metabolism in our body. Now, these are an example of naming, but they also show you something interesting about sugars, which is that many carbohydrates are isomers. And we're going to talk about the different types of isomers that exist within carbohydrates. Here you can see that both of these sugars have 3 carbons in them. And in fact, their chemical formula is identical. That means that they are an isomer. But you can also see that between glyceraldehyde and dihydroxyacetone, their arrangement of the atoms in their structure is very different. One is an aldehyde, one is a ketone. So these two sugars are called constitutional isomers, meaning, again, they have the same chemical formula, but a complete different rearrangement of the atoms and the bonds in the molecule.